This lesson will be about how DNA copies itself, a process called replication. Here we see the DNA molecule in a space-filling configuration. Notice that the blue atoms, those represent the nitrogen bases. Those are in the center of the molecule. The phosphates and sugars are on the outsides. So here we've got a diagram of a nucleus of a eukaryotic cell. The nuclear membrane on the right is labeled with a nuclear pore. The pores are openings into and out of the nucleus surrounded by gate proteins that regulate what can enter and leave the nucleus. Cytoplasm outside and the nucleus inside. Let's take a minute and label some of the structures in the nucleus. Chromosomes are made out of DNA wrapped around proteins called histone proteins. When it comes time for DNA to copy itself before a cell divides so that the new cell has instructions as well, then a series of enzymes help to uncoil the DNA from the chromosomes, untwist it from the double helix, break the hydrogen bonds that hold the base pairs together, and then insert new complementary base pairs. We're going to start with the enzyme that uncoils the DNA from the chromosome. We'll use our little enzyme symbol here to show that what it's doing is removing the histone proteins, unwrapping the DNA from the chromosome. A second enzyme, called Helicase, untwists the double helix. We'll have two Helicase enzymes, one at either end of where the DNA is going to start copying. Now, the place where the DNA starts replication is called the origin of replication, or the replication origin. And in a eukaryote cell, though there are several 100 to even 1,000 replication origins on the different chromosomes. In prokaryote cells, there's a single replication origin. Shown here are two in this diagram. After the DNA has separated the two halves and the hydrogen bonds holding the base pairs together are broken, single-strand binding proteins attach to the strands to prevent them from re-annealing together, recombining together. Shown here are the single-stranded binding proteins. The untwisting of the double helix at one end of the replication origin causes the double helix to twist even tighter further down the line. You can imagine that as Helicase here untwists the DNA further down this direction, it's twisting tighter, and the same as this Helicase untwists, it keeps twisting tighter that direction. So there's another enzyme called topoisomerase, and the job of that enzyme is to break the sugar phosphate backbone and allow the one half of the DNA strand to untwist around the other, and then it will re-seal that half so that it keeps the DNA from super twisting and getting gobbed up. So we'll draw topoisomerase here as well. The most important of the enzymes is the one that actually builds the DNA, and we're going to call that DNA polymerase because it polymerizes or builds DNA. We're going to represent that with a kind of a donut-shaped enzyme here. Now, remember that the two strands of the DNA are parallel but running in opposite directions, so they're an anti-parallel configuration. And the reason that's important is that the enzyme DNA polymerase can only build DNA in a certain direction. It can only build from the 5' to 3' direction. Remember, the deoxyribose sugar has the carbons numbered, with number 1 being the one that attaches to the base, number 3 attaches to a hydroxyl group that binds to the next phosphate group in line. So it turns out that this enzyme can only build in a certain direction. So we'll label that direction of these strands here 5' to 3'. Notice if one strand is 5' at the left and 3' at the right, the complementary strand will be the opposite, 3' at the left, 5' at the right. And when we build the new strand, it will need to be built opposite 5' to 3'. Now, the enzyme DNA polymerase would love to be able to just bind on anywhere and start building DNA, but unfortunately, the enzyme needs a double-stranded area to know where to begin. So in actuality, the first enzyme that will hook onto the DNA strand is called primase, and it can build short pieces of RNA that bind to the DNA, called primers. We'll use blue to represent RNA instead of DNA. So the new strand have an adenine that binds to the thymine, But instead of thymine, RNA doesn't have thymine, it has uracil, a U instead. Guanine still binds to cytosine. So imagine that this is a short piece of primer. Now the DNA polymerase enzyme can bind on and begin copying or building new DNA. We'll use a different color to represent the new DNA. And remember, DNA has T's, or thymines. Now, at this point, when the DNA polymerase enzyme reaches this position, does it just have to stop? No, because as it's moving this direction in building new DNA, the topoisomerase, the helicase, and the uncoiling enzymes are all moving down the chromosome in this direction, uncoiling, untwisting, and unzipping the double helix. So by the time the DNA polymerase enzyme reaches here, more DNA has unwound and it just continues, it just keeps continuing. A problem occurs on the other half of the DNA because it's in the wrong direction. If it's going to be built 5' to 3', then the primase and the DNA polymerase enzyme have to go the opposite direction. The problem is the entire strand of DNA is not untwisted yet, so the primase has to start partway through the strand. Let's imagine primase lands here to begin building the primer and moving this direction. There's the primer. Now DNA polymerase can find the correct location. It lands and begins moving this direction as well, building DNA. Now when DNA polymerase gets to the end here, because the rest of the DNA is still twisted up and it can't build further that direction, it falls off, moves back this direction to where it finds another primer that has been built, then begins copying new DNA this direction as well, 5' to 3' always. Once it gets to the place where a primer already exists, it falls off, moves further back, and by this time more of the DNA has been untwisted and unwound. So it can continue to build in these short segments, these discontinuous segments in the 5' to 3' direction. Now the other one on the other DNA polymerase on the other strand just continuously moves this direction all the time. So we call the one strand of the DNA the leading strand. It's built continuously, it's a little bit faster, and the other strand that's built in segments is called the lagging strand. But at the end, they both are the same. Now, of course, eventually we don't want these pieces of RNA, these short pieces of RNA called primers, in the DNA. So, you guessed it, another enzyme comes along and removes those primer pieces and replaces them with DNA. So you can see by the end, both strands of DNA should be identical, assuming that the DNA polymerase enzymes don't make any mistakes and the other enzymes don't make any mistakes as well. Now, of course, in a perfect world that would always happen, but of course the interesting things happen, and occasionally mistakes happen. Now these enzymes are incredibly accurate, really incredibly accurate, but they do make mistakes. It's estimated that, or all 6 billion bases of human DNA in a single cell, that 1 or 2 mistakes happen every time the DNA replicates. This is an image of DNA polymerase. In this view, and you can see that it has the hole in the center. The DNA fits through the center, and then it clamps down and travels along the DNA strand, building the new DNA. Now if we change the color to show the different polypeptide change, you can see that it's actually built out of 4 different polypeptides. And that little red one, very interestingly, you can think of that as, in a way, a spell checker. It is at the very trailing edge of the enzyme, and if a mistake happens, it often catches that because the molecule will be the wrong shape, nitrogenous base that's been fit into the DNA strand, and it will stop the enzyme, cut out the wrong base, and replace it with the correct one. So this is an amazing little enzyme, this DNA polymerase. This process of replication begins at multiple origins along each chromosome, and the enzymes follow each other through, always building the DNA from the 5' to the 3' direction, and at the end of the process, which can take several hours in a eukaryotic cell, the result is 2 new strands of DNA where originally there had been only 1. Now if you notice, each strand is composed of 1/2 that's old DNA and 1/2 that's new DNA, so it's a semi-conservative process of replication. And this is how DNA copies itself. Now again, this only happens once during a cell's lifetime. Just before the cell divides, it will replicate the DNA.